Hello! In this video, I'll be talking about the relationship between epigenetics and nutrition. Epigenetics is the study of how environmental factors like nutrition can influence the expression of certain genes without changing the actual genetic code. Instead of changing the actual sequence of a gene, epigenetic changes affect the way genes are read. There are three kinds of epigenetic changes I'll be talking about in this video. They include histone modification, DNA methylation, and non-coding RNA modifications. Histones are the proteins that DNA wraps around to form a chromosome. The way in which histones are shaped can make certain genes accessible or inaccessible to transcriptional factors in RNA polymerase, which creates mRNA to be translated into proteins later. Dietary factors can trigger histone modifications and gene expression in this way. DNA methylation and acetylation are other types of epigenetic changes that can change the way DNA is read. Methylation is the addition of a methyl group to a section of the DNA, usually in the promoter region of a gene. The addition of a methyl group to a section of DNA can silence a gene or promote its transcription. The effect it has on different genes varies. DNA acetylation is the addition of an acetyl group to a segment of the DNA. Acetylation can cause a methyl group to leave a DNA segment in a process called demethylation and the relaxation of a histone protein to make the DNA more accessible to RNA polymerase and transcription factors. Another way gene expression can be changed without changing the actual code of the DNA is through non-coding RNAs. Non-coding RNAs are molecules that alter the way genes are read, or if they're even read at all. One type of non-coding RNA, called small nuclear or siRNA, can cut and paste together sections of transcribed messenger RNA so that it codes for a specific protein. This is how we are able to make thousands of types of proteins with a relatively small genome. It's not that we have the code for every single protein independently laid out in our DNA. Instead, siRNAs arrange segments of DNA that make a set of instructions for a specific protein when it's needed. Because these alterations are done on the messenger RNA and not that DNA itself, this is considered an epigenetic change. Another type of non-coding RNA, called microRNA, binds to messenger RNA to keep it from being translated into a protein. It essentially puts the messenger RNA on hold until it's needed. Dietary components are capable of changing gene expression by acting as nuclear receptors that can bind directly to the DNA or interact with transcription factors that can initiate transcription. Several studies have explored the effects of nutrition on epigenetic changes. One study on the agouti protein, which determines coat color in mice, found that pregnant brown mice fed a high methyl diet had offspring that were yellow instead. The agouti protein was methylated in the offspring, causing a color change. More interestingly, even when the offspring were fed a normal diet, their offspring continued to have a yellow coat. This means that epigenetic effects can be transgenerational. Another study on rats found that severe caloric restriction in pregnant rats led to more methylation in genes that were associated with metabolism in their offspring. For example, there was more methylation in the INS2 gene, which controls the production of insulin. This is extremely important, as it suggests that maternal nutrition can influence the metabolism of their offspring. One real-life example of how nutrition can affect epigenetics is the Dutch famine during World War II. In the last year of the war, Nazis staged a blockade that cut off the food supply to the Western Netherlands. During this time, millions suffered from malnutrition. Later studies found that children of the women who were pregnant at the time had higher rates of heart disease, altered glucose metabolism, and changes in methylation patterns. Specifically, they had less methylation at the IGF-2 gene, which regulates growth. If not regulated properly, the IGF-2 gene can contribute to cancer growth. Additionally, They had more methylation in genes that controlled satiety and cholesterol metabolism. These findings suggest that prenatal malnourishment can affect metabolism later in life, even when environmental conditions change. Several diseases have been linked to nutrition and epigenetics. However, in this video, I'll be talking about cancer, cardiovascular disease, metabolic disorders, and mental health disorders. Cancer is a disease characterized by the uncontrolled growth of cells. There are several factors that influence the proliferation of cancer, like genetics, environmental factors, and age. Throughout our lives, we are constantly replicating our DNA. This can lead to an accumulation of mutations that result from mistakes in the DNA replication process. This is why people tend to get cancer later in life, as they have accumulated more mutations. Cancer has also been linked to abnormal methylation patterns in different genes. Hypomethylation and hypermethylation at specific genes have been associated with the growth of tumors. For example, hypermethylation in tumor-suppressing genes like VHL and P53 have been associated with the proliferation of cancer cells. However, not enough methylation at genes that promote cell growth like BRCA1 has also been associated with cancer. Another type of disease related to epigenetics is cardiovascular disease. Inflammation, oxidative stress, and increased methylation at certain genes have been associated with cardiovascular disease and aging in general. Dietary factors like flavonoid and polyphenol intake, as well as slight caloric restriction later in life, have been shown to reduce methylation patterns leading to inflammation and oxidative stress. Some cardioprotective flavonoids include naringenin, luteolin, and quercetin. As we saw in the example of the Dutch famine, epigenetic changes brought on by nutrition can have lifelong effects on metabolic processes. Several metabolic diseases associated with epigenetics and nutrition include diabetes, osteoporosis, hyperthyroidism, and obesity. Another study on mice found that pregnant mice fed a low-calorie diet had offspring that had glucose intolerance, increased fat cell production, and larger fat cells. When these offspring were fed an even lower calorie diet than their mothers, they had better glucose and lipid metabolism. This is really interesting because it suggests that these offspring were metabolically programmed in utero to survive in an environment with little food. When brought into an environment that didn't match their prenatal environment, they had worse glucose and lipid metabolism. The effect of nutrition on epigenetic changes that can contribute to mental disorders has also been studied. Folic acid is a type of metabolized B vitamin that can act as a methyl donor. Folic acid deficiency during the prenatal period and early brain development have been linked to a higher risk of developing depression later in life, as well as cognitive and motor delays. Choline is another dietary factor that may affect mental health through epigenetic modifications. Choline is an important cation found in many foods and is needed for the production of an important neurotransmitter, acetylcholine. It can also affect methylation patterns. In one study, infants whose mothers were given a choline supplement during pregnancy had increased methylation in genes. Now that we've talked about some diseases that are linked to epigenetics, let's talk about some dietary components that can change the way our genes are read. One interesting vitamin involved in epigenetics is vitamin D. Vitamin D binds to a receptor inside the nucleus called the VDR receptor and can regulate gene expression by interacting with transcription factors. Interestingly, vitamin D can regulate gene expression in direct and indirect ways. Vitamin D can indirectly affect gene regulation by binding to proteins in the nucleus that interact with histone remodeling and methylation proteins. It can also target and initiate transcription of genes that code for proteins that can modify the chromatin shape. Flavonoids are another dietary component that may contribute to epigenetic changes. Flavonoids are molecules made by plants that give them their color and help them defend against pathogens. They're also known for their antioxidant and anti-inflammatory properties in humans. As we know now, oxidative stress and inflammation have been linked to cancer and cardiovascular disease. Flavonoids may also prevent cancer by preventing the buildup of mutations that cause cancer by turning on genes that make proteins that fix mistakes in DNA replication. One flavonoid in particular, naringenin, can alter gene expression by influencing microRNA activity. As I mentioned before, microRNAs are a type of non-coding RNA. Some studies have shown that naringenin can increase the expression of proteins that protect cells from oxidation and cell damage, like manganese superoxide dismutase and glutathione peroxidase 2, which may prevent the buildup of mutations that can cause cancer. B vitamins like folate, B6, and B12 control the production of a molecule called S-adenosylmethionine in the body. S-adenosylmethionine acts as a methyl donor and is capable of silencing or activating certain genes. Overall, epigenetic changes can have an enormous impact on virtually every aspect of our health. It's important to continue studying how our diet can affect the expression of our genes, so we can better understand how diseases like cancer and heart disease develop, and how we can better prevent and treat them. Thank you for watching this video, and please feel free to look through my resources, as well as conduct some research of your own on how our nutrition can affect our epigenome.